Guys, it's Steve. I figured I'd just do a little bit of walking around this Cosmic Variance Navigator with you guys, just in case you had some questions. So first of all, what is Cosmic? So if you look at this tab here, you can see this is the catalog of somatic mutations in cancer. This is from the Sanger Center. So what they've done is they've compiled all of the single nucleotide variants related to cancer. So because they had control tissues as well as cancer tissues, they could select out the somatic mutations that are specific to the cancer. So this is great. This is exactly what we're after. The trouble is that you're effectively looking at a table with 17 million entries. So how do you find something interesting out of that? So what we've done is we've built a tool that lets you select a range or a gene and displays where the most mutations are occurring. So effectively what we have in the main plot is a histogram of where the coding and non-coding variants are lining up with the genes. So you can see in the gray boxes, these are the gene diagrams. You've got some arrows to show direction of coding for that gene. And then you've got green lines for the coding variants and a blue line for the non-coding variants. And then you can easily spot where spikes are occurring in that gene, so where most cancer variants are for that gene. So beyond that, what we can do is we can change the genomic range. If you, for example, look here, we can change the range in the HG38 assembly that we want to plot in the histogram. And we can also change the window size for how that histogram gets binned. So by default, it just goes and plots INHBE next to GLE1, so it's plotting that whole range. And it's defaulting to a binning size of 100 base pairs, which means that every one of these levels is spanning 100 base pairs of that gene. So we can change that to whatever we want. Obviously, the smaller it goes, the more data. That's going to be a little bit harder to interpret, but it defaults to 100, and we can change that if we want. So the other thing we've done is, because maybe we don't have all the locations memorized, I've pre-programmed in some of the genes of interest for us. These are the hedgehog genes: GLE1, GLE2, GLE3, Patch1, Smoothen, and Sonic Hedgehog. So if we want to jump to another gene, for example, Patch1, all we have to do is select it and then hit Submit. You'll notice that it's connecting to database, and then it replots this diagram. So again, we get a nice diagram of what the gene is and what direction coding is, and we now see all the spikes for coding and non-coding variants for that gene. And we can change the window size as well if we want, maybe make it a little bit bigger. So we can change that to 300. We just hit Submit again. It'll reconnect to the database, replot the histogram, and then we can see where these spikes and variants occur. But we're not just interested in where the hot spots for spikes are. You can see it just updated. We're not just interested in where the hot spots are, we're also interested in what types of cancer. So if you click on the histogram, what we get below it now is an updated view of what types of cancer we're looking at. So for example, here we've broken it out based on primary site as well as histology. So when you hover over a plot, what you get is the site where those cancers occurred, the number of cases, the p-value on whether or not it's significantly changed from the entire database, and then how many of those variants are in what locations and in what histology in that location. So what we see is that 36% about of the 300 variants in that window are adenocarcinoma in the large intestine. And we can see the p-value is effectively rounded off to 0, meaning that's a very significant change from the rest of the database. So this donut plot on the left is for the window that we clicked in. The plot on the right is all the plot range, so all the variants in the entire plot range. And the bottom is all of the coding variants across all of Cosmic. So that's all 5.2 million variants and how the distribution breaks out. So we've calculated p-value based on a binomial variation, two-tailed. So we're looking if it went up or went down. And in this case, it looks like it went up, because here you see it's 36%. For the entire database, it's only 20%. So they're saying that that is, we're estimating that that is a significant increase in large intestine-based cancers. Now, if we need more data than that, we can go down at the bottom, and what we have is all of the original data from Cosmic displayed in a table. And in typical HTML format, what we're looking at is we can change how many entries we're looking at in the table. And across the top, we can sort based on any of these entries, based on primary site ID tumor, whatever you want to sort against. So any data that we couldn't graphically represent that is original to Cosmic shows up in this table, because this is the data straight out of Cosmic. So that's the table at the bottom. And then you can click through the pages here on the bottom if you want to go to the second page. There you go. And if we want to flip over to the non-coding side, you just click the tab. There's the non-coding side. So obviously, since we're in one of the exons, all of the variants should be in coding, and that's what we see. So we see 0 non-coding events and all of the coding events. So again, we can flip around to another gene if we want. Go over to smoothing, hit submit. You can see the window size stayed at 300. This is a much smaller gene, so you can see that the histogram looks a little bit chunkier. So we can go back and change our window size, something a little bit more interpretable. Change that to 50, hit submit. There we go. Now we've allowed you to enter in any range you want manually. It's going to interpret it based on chromosome, colon, position, dash, and then position end. So that's exactly the format you get out of UCSC's genome browser. So if there were a gene that you weren't sure the position in HG38 assembly, you can type the gene in genome browser and then effectively just copy and paste that location right there. Update our window size, hit submit. It's going to show you a plot range for that gene. So how this is working is we're effectively using R, the statistical environment, as the server backend. And some of the packages in R allow us to use a browser as the front-end interface for going through all this data. So those packages are Shiny for all the HTML building and CSS styling, and then Plotly for doing all the graphing of the donut charts and the histogram. So using all those plots together, we've got a nice manual environment where we can cruise through this data looking for hotspots in genes of interest without having to dig through millions of entry in a data table. Great. Hopefully that explains everything. Hope you guys enjoyed it. Thanks a lot.